What we have here is a very crude drawing of a neuron. I'm going to quickly outline the particular parts just as a reminder. If you can see the little pencil, I am now touching the dendrites. This is the cell body. This is the nucleus. This is the axon. Now around the axon that I have not drawn here are like big bubbles of fatty tissue called myelin sheathing that cover this axon. And the thicker the myelin sheathing, the thicker the signal, or the faster the signal can move. And now we're traversing to the end of the neuron. This is the axon terminal. These are all axon terminals down here. And what you can't see because it's too small is on those axon terminals there are synaptic vesicles. These synaptic vesicles hold neurotransmitters. Those neurotransmitters, when the cell is told to fire, they release the neurotransmitters into this synaptic gap, this gap between the two neurons. Now, this gap is not filled with nothingness. The gap is filled with cerebral fluid. And cerebral fluid it's much like salt water. It's not exactly like salt water, but for our purposes, think of it, it's kind of like salt water. Now, how this neuron is going to talk to this neuron is when this neuron is told to fire, the synaptic vesicles, which are too small for me to show you here, that hold the neurotransmitters, pop and release the neurotransmitters into the cerebral fluid that is occupying this synaptic gap. This is the equivalent of, if you want to think of it this way, think of an eyedropper that somebody's filled with red food coloring. And if I was to take that eyedropper of red food coloring and put it in a glass of water and then press the plunger, if you could watch that in slow motion, what you'd see is coming out of the tip of the eyedropper would be a very concentrated red dye that would, disperse amongst the water, and within seconds the entire glass of water would now be pink. That's sort of what happens here when the neurotransmitters are released into the synaptic gap, because the neurotransmitters, those chemicals are released into this fluid. And now they're going to be occupying this local fluid right here. Now, the neurotransmitters have a chemical signal. They have a chemical signature, and they have a particular shape, their chemical shape. That chemical shape fits perfectly into bind sites on the dendrites of this next neuron, right where the pencil is now. Those bind sites they act as little locks, and the neurotransmitters are little keys that have specific chemical signatures, in other words, a shape that fits into those locks. And enough of those locks or bind sites are opened, and it tells this next neuron, fire. And that's how one neuron talks to another neuron.